Hi all, welcome to part 3 of endocrine. I'm going back to the previous slide, the slide that ended the last video. So here we have a cell. Let's start with the one on the left here, and it could be any cell. It could be a skin cell, it could be a muscle cell, it could be a cell from the lung. It's any kind of cell that needs glucose if it's going to survive. And this one also has an insulin receptor, as all cells do, and in the presence of insulin, it will be able to take in this glucose. But in this diagram here, no insulin is present. So the transporter of glucose, the GLUT4 here, is not embedded in the membrane. It's inside the cell, so it's not going to do any good there until insulin is released, attaches to this receptor, and allows this GLUT4 transporter to actually transport. Okay, so we're calling this the resting cell. And here they're telling you it's either skeletal muscle or adipose cell. It could be anything. So now what happens once that pancreas has released that insulin that we saw in the previous video? Okay, now it's released insulin. What happens here in step 2 in part B? Now that insulin is going to bind here to that receptor. So the little yellow ball is the insulin. It bound to the receptor. And it's going to trigger a cascade of signals. And the outcome of that cascade is that GLUT4 transporter, different from the other transporter we talked about for glucose, that the CGLT, that would have also sodium present. This one will do it on its own. So now, if I bring this over to the membrane, insert it, what's going to happen is you now have a channel that will allow that glucose to enter. So look at these little purple balls are now able to enter. Whereas back here, because that insulin receptor did not have that insulin attached, I could not move that transporter yet to the membrane, allowing the glucose in. So this is a receptive cell, a cell that has, under the effect of insulin, allowed itself to import all this glucose into the cell. So that's the effect that insulin is having upon these cells, allowing them to take in more of that glucose. I wanted to just highlight there that what was used was a GLUT receptor, which is a receptor for glucose. And in the last chapter, as I said, we saw the CGLT receptor, which was the one that transported along with sodium. I'm telling you, that's how cells communicate with each other, right? One cell from the pancreas is saying, okay, this is the signal, go for it. Now you have to take that sugar in. Now, that action is fine. It's the cell is taking in the glucose. But that also must stop because it can't forever take in the glucose. So that action needs to stop. Also, what has to stop is the release of the actual insulin from the beta cells of the pancreas. So how this works is that hormones in the bloodstream are degraded. So that when you release that insulin, it works for a little bit, it does its job, and then it's going to degrade or break down and no longer be active. So we want to know that. We want to know how long does it stay active. It's a hard thing to study because you want to look at it and you see that it's present and then you see that it's gone. And you don't exactly know at what point it was gone. So it's easier to study it in terms of a half-life. You want to know when half of it is gone. And then you want to know when the half of that half is gone. And that's how we study the effect of hormones and of pharmaceuticals too. You take Advil and you want to know how long is it going to last? How long is it before I only have half of that acetaminophen in me? Not acetaminophen, that's Tylenol. Any molecule, you want to know how long it's active. So when it's naturally occurring, it's kind of short. It's around 5 minutes. It fluctuates 4 to 7 minutes, naturally occurring. Synthetic, when we're talking about insulin, that wouldn't be very good for an individual to have the effect of that synthetic insulin lasting 5 minutes, and you'd have to retake the insulin all over again. That would not be very practical. So the synthetic one is anywhere from 2 to 6 hours, depending on these different brands. So much better when it's exogenous, when you're taking it as a medication. So there is an enzyme in the blood that will break down that hormone over time. If you define half-life, then it's the time it takes for a substance, such as a drug or a hormone, to lose half of its pharmacological activity. And it's just easier to measure that way, and that's why we talk about half-life. This is a diagram of that, where you would have the concentration of whether it's your insulin or some Advil again over time. So you could measure that and know, this is declining, and then you measure it at a certain point in time, and you see that you have about half of that component at 400 seconds here. And then you'll want to know, okay, I have that amount. I have 0.8 concentration. How long will it take until I have 0.4, until I have that again? So this is taking half of that. And now I have 0.4. How long will it take until I have 0.2? And so on and so forth. And so you create a curve here that will, this curve here is showing that breakdown and how long it takes to break down these hormones in the blood. We're going to stop there. Continue on the next mini lecture. Thanks for listening, guys.